Welcome back to MLT 120. In today's lecture, we're going to be covering hematopoiesis and erythropoiesis. Topics are going to cover how blood cells come into existence and how red blood cells mature. So to start off this lecture, let's first define hematopoiesis. It is the production of blood cells. So let's break the term down. Hemato means hematology. Poiesis means to produce, so we're producing blood cells. The term hematopoiesis includes all three of the blood cells that we have learned that are in our blood, which includes our red blood cells, our white blood cells, and our platelets. So where do you think hematopoiesis takes place? It primarily takes place in the bone marrow. And once the cells are mature, our blood cells are mature, they leave the bone marrow and they enter circulation. And once they enter circulation and are going throughout the whole body, the blood becomes known as peripheral blood. So the blood throughout the body is peripheral blood. But we have two types of bone marrow. We have yellow marrow and we have red marrow. The red marrow is the cell producing or the active marrow. And then we have yellow marrow that is more known to be inactive and is composed primarily of fat. We just learned that the site of hematopoiesis or blood cell production in adults is in the red bone marrow. But that's not the case for the developing embryo or fetus or our newborns. So where does the development happen for our blood cells in the fetus? Well, in the first stage, hematopoiesis begins in the yolk sac. So what is the yolk sac? It develops inside the mother, inside the uterus, and provides the embryo with nourishment and also helps circulate gases for the first 4 weeks of life. This is known as the mesoblastic phase. Next up, at around 4 to 7 weeks, hematopoiesis occurs in the liver. This is known as the hepatic phase. At about 4 to 5 months old, the bone marrow starts to develop your red blood cells. This is known as the medullary phase. Sometimes you'll hear it referred to as the myeloid phase. The very first type of blood cells that develop are our red blood cells. And the first form to develop is known as the erythroblast. A little bit later on in the lecture, we'll be talking about the maturation sequence of the red blood cell and the different forms. Finally, once the baby is born, hematopoiesis occurs in all bone marrow. And this is because when babies are born, all marrow is red marrow. Remember, we said red marrow is active marrow and it's cell producing. Okay, where adults have red and yellow is inactive, babies have all red marrow. Yellow marrow usually doesn't develop in children until around the age of 2 to 5 years old. So in adults, we know that blood cells are produced in the red marrow, but where are the sites in the body that contain red marrow? Some of those areas include the skull, the sternum, the proximal ends of the long bones like the tibia and the femur, the vertebrae, the ribs, and the iliac crest. If you'll look here on the image on this slide, you'll see this is an example of a long bone in the body. You see the red marrow is at the proximal end of our long bone, and then you have the yellow marrow down in the shaft of the long bone. And remember, our yellow marrow is the inactive marrow, so it wouldn't be producing any blood cells for us. Sometimes in adults, there's diseases or conditions that can cause a failure of bone marrow production, or it can affect the production of our blood cells. So the body can produce blood cells through what we call extramedullary hematopoiesis. And this can happen in rare cases, and we can produce blood cells through the liver and the spleen. We're going to learn about the theory of blood cell origin next. This is a theory where the blood cell origin is described as an early stem cell, and there's a maturation sequence that happens within the bone marrow. And once the cells mature in the bone marrow, then those blood cells are released into the blood. And we're going to talk about it a little bit more in detail on the next slide. But you can see here is an example of our bone. And of course our marrow is inside of our bone. And all this maturation sequence of all of our blood cells, it doesn't matter if it's a red blood cell, white blood cell, or our platelet, everything's going to mature inside the bone marrow before it's released into the peripheral blood. In the theory of blood cell origin, all blood cells, whether it's your red blood cells, white blood cells, or your platelets, all those blood cells are derived from the pleural potential stem cell. In some texts, you'll hear it referred to as the multipotent progenitor stem cell. This is an uncommitted stem cell at the time of development, and it can become, like I said, any of the three types of blood cells. It can become a red blood cell, it can become a white blood cell, or it can become a platelet. It just depends on how it matures in your bone marrow. Before we move on to the second bullet on this slide, let's first define what cytokines are. Cytokines are things that are produced, or substances that are produced by the cells that stimulate other cells to produce things or tasks. So keep that in mind. So let's look at that second bullet. In response to cytokines, the pleural potential stem cell will develop to either a committed myeloid stem cell or a committed lymphoid stem cell. So your pleural potential stem cell is uncommitted, but in response to a stimulus, the cytokines, it's going to be split off and either become a myeloid stem cell or a lymphoid stem cell. So we're starting to divide off and kind of decide what we're going to be as far as what type of blood cell we're going to be. The myeloid stem cell can mature and differentiate and become either a red blood cell, a granulocytic white blood cell, a myelocyte, and a platelet. And I know we haven't talked about all those in detail yet, but those are the possibilities for the myeloid line. For the lymphoid line, if it goes and develops to the lymphoid side, the stem cell can differentiate and become a lymphocyte, and it can be a T or a B lymphocyte, which is also referred to your B cell or your T cell. So let's do some trivia questions based on the information that we've just learned. I would encourage you to pause the video on the questions, try to answer them yourself before you go on to the next slide to get the answer. So our first question is, a mature red blood cell is derived from which committed stem cell? The answer is the myeloid stem cell, because in the maturation sequence, the red blood cells come from the myeloid line. Our next question is, all cells are derived from which uncommitted stem cell? The answer is pleural potential stem cell. We learned that the pleural potential stem cell has the potential to become any of our blood cells. Our third trivia question is, a mature lymphocyte is derived from which committed stem cell? And of course the answer is our lymphoid stem cell. We learned that only our lymphocytes come from our lymphoid stem cell side. Now we need to discuss and learn what erythropoiesis is. The term erythro means red, and we learned earlier in the first lecture that the red blood cells are also referred to as an erythrocyte. So erythropoiesis refers to the development of red blood cells. So there are several steps that the red blood cell must go through to mature and become the red blood cell that we're familiar with. So you'll see here in the diagram the several steps that the red blood cell must go through to mature to become the nice little donut shape or biconcave dish shape that we're familiar with as a mature red blood cell. Before we get into the different steps of the maturation sequence for the red blood cells, you need to know there are actually three different naming systems or three nomenclature systems for erythrocyte maturation. Now we're only going to learn two. Don't ask me why there's three naming systems. I'm sure I say it's just to make things confusing, but we're only going to learn two of the naming systems. We're going to learn the normoblastic and we're going to learn the ruboblastic. And you can find a chart on page 63 in your textbook. And the erythrocyte production and maturation begins, of course, in the bone marrow. Okay, now we're going to go over the very first step in the maturation sequence for the red blood cell. And the way that I've got this set up is for each step we've got the two different names for the naming sequence up here at the top. On the left-hand side, we've got the normoblastic naming system, and on the right-hand side, we've got the ruboblastic. So for this first step in the maturation sequence for red blood cells, the normoblastic, the first step is the pronormoblast, but for the ruboblastic system, the first step is known as ruboblast. So those can be used interchangeably for my purposes, but no, it's a different naming system. Okay, you can find a image on page 19 in your atlas. So I would encourage you to pull out your atlas at this point and follow along in your atlas as you're watching this part of the video. But like I said, this is the first stage of cell maturation for your red blood cells. In this step for the red blood cell, the cell can divide. The nucleus of the red blood cell is oval or round to slightly oval. There's very fine nuclear chromatin. And in the different steps of the maturation sequence, we'll be talking about the end-to-C ratio, and this is something known as the nuclear to cytoplasmic ratio. And in different steps, there'll be different ratios for each step. But in this first step of maturation for red blood cells, the end-to-C ratio is 8:1, which takes up most of the cell, which means the nucleus is taking up most of the cell. And also there's nuclei that are present within the nucleus. As far as the cytoplasm of the cell is dark blue, and you can see the Golgi apparatus. Again, I strongly encourage you to pull out your atlas and look at the images in your atlas that go along with this step of maturation of the red blood cell sequence on page 19, so you can see what the nucleus looks like, what the cytoplasm looks like, so you can compare what this step looks like versus the next step coming up. The next step in maturation of red blood cells is the basophilic normoblast, or you can also refer to it as the prorubrocyte step. You can look on page 21 in your atlas to see an image of what the step looks like for the red blood cell. In this step, your red blood cell can still divide. As far as the nucleus is round. The chromatin, or the nuclear chromatin, is now slightly condensed, so it should look a little bit thicker where it was thinner in the first step. You may see nuclei, and the end-to-C ratio, or the nuclear to cytoplasmic ratio, is now 6:1. So that nucleus is not as big as it was in step 1, so it's not taking up as much space in the cell. For the cytoplasm is dark blue, which means there's more RNA present, and there's not as much hemoglobin at this stage. If there was more hemoglobin, there would be more of a pinkish or red color. Again, please make sure you're looking at your atlas to see images of each of these steps. The third step in the red blood cell maturation sequence would be the polychromatic normoblast, or you could say the rubrocyte stage. And again, the polychromatic normoblast is referring to the third stage in the normoblastic naming system, and the rubrocyte is referring to the third step in the rubroblastic naming system. Check out page 23 in your atlas to see what this step looks like for the red blood cell. This is the last stage the cell can divide. In the nucleus will appear round. The chromatin will be even more condensed than the previous step. You will not see any nuclei, and the end-to-C ratio is now 4:1. So that nucleus is shrunk down even smaller. It's taking up less of the cytoplasm. The cytoplasm, the color is now a gray-blue because hemoglobin is finally being produced. We usually represent hemoglobin with this HGB. The majority of the cell's hemoglobin is produced in this stage. This is important, that's why it's underlined. The majority of the cell's hemoglobin is produced in this stage. Because in this stage you're still seeing blue from the RNA and some pink from the hemoglobin, this is known as polychromatic. Poly is meaning many colors. The poly is the many, and chromatics referring to colors. Alright, the fourth stage in the red blood cell maturation sequence is orthochromic normoblast, or you can refer to it as the meta-rubrocyte stage. Please look at page 25 in your atlas to see the image of this stage. The cell can no longer divide. The nucleus is small at this point. The chromatin is very condensed. We call it pycnotic. Pycnotic is just a term that means that the chromatin is clumped together. And the end-to-C ratio at this point is 0.5:1. So we can see now by that ratio that that nucleus is really small. And at this point, we also refer to the red blood cells as NRBCs. So you might want to write that down in your notes, NRBCs, and that means a nucleated red blood cell. It basically looks like a red blood cell with a nucleus in it. As far as the cytoplasm, the color is more of a salmon color, so it's more of a pinky color. You have less blue because of the increase of hemoglobin in the cytoplasm. Remember I said when you have hemoglobin, you're going to have more of a pink or reddish color. So that's what's happening in this stage. But please make sure that you're looking at your atlas and seeing the images of each stage of maturation. The next stage in maturation is the polychromic erythrocyte stage, or the reticulocyte stage. This is on page 27 in your atlas. Please check out that image. At this stage, the cell is released from the bone marrow into the peripheral blood. So we're still not completely mature yet. We're still maturing, but it can be released into the peripheral blood. The nucleus has been extruded, so it's no longer present. So we don't see the nucleus at all. The color of the cytoplasm is a pinkish blue because while it mostly contains hemoglobin, it still has some residual RNA and ribosomes in the cytoplasm. And we must use what we call a supravital stain to see the reticulum or that ribosomal material in order to actually ID it as a reticulocyte or at this stage of development. One other thing I want to mention before we move on to the last stage of development is a lot of times you'll hear lab techs just refer to your polychromic erythrocytes as just polys for short. So if you ever hear that term, just a poly, they're referring to your polychromic erythrocytes or your reticulocytes. This is our final stage of red blood cell development. This is our mature red blood cell, the erythrocyte. Remember I said you can use those terms interchangeably, red blood cell and erythrocyte. So you want to see page 29 in your atlas to see an image, but we also have an image here on the slide of a mature red blood cell. The cell at this point is fully mature and it's out in the peripheral blood. You will not see any nucleus. The cytoplasm is a salmon pink color, and you'll have an area called the central pallor because the cell is what we refer to as a biconcave disc shape. So let's look at this image. I've been referring to this as kind of a little donut, but it's not a true donut. You don't have a hole through it. This little divot here, this divot is known as your central pallor. This little divot is the central pallor. And the whole cell itself is known as a biconcave disc shape. Okay. Notice that the mature erythrocyte is smaller than the previous polychromatic erythrocyte stage. The normal size of a mature red blood cell is only about 6 to 8 microns. And the average lifespan of your red blood cells is about 120 days plus or minus 10 days. So that averages out to about 110 to 130 days. Now let's talk about the red blood cell function or the erythrocyte function. What does the red blood cells do for the body? The primary function is for the erythrocyte to carry oxygen to the tissues. So it carries oxygen all around the body. So cells and tissue must have the oxygen to produce energy or ATP. Remember, we refer to energy as ATP to survive. Without the oxygen, those cells and tissues would die. Also, the hemoglobin molecules in the red blood cells carry oxygen from the lungs to other tissues. Another function is that red blood cells also pick up carbon dioxide from the tissues and transport it to the lungs for us to breathe out. This state of hypoxia, which is low oxygen, triggers the bone marrow to increase the production of red blood cells. Okay, so we just learned that if there's a state of hypoxia in the body or low oxygen, the body kicks off and wants more red blood cells. Well, there's a hormone called erythropoietin, or EPO for short, that is produced in the kidneys. When oxygen is low in the body, this is triggered and the kidneys produce more and release more EPO or erythropoietin, which stimulates the bone marrow to produce more red blood cells. So once the body wants more red blood cells, the hormone erythropoietin that's produced by the kidneys tells the bone marrow, hey, make more red blood cells. And the effects of erythropoietin increases the number of stem cells, it increases hemoglobin synthesis. It decreases the maturation time. So all those steps that we just looked about for red blood cell maturing, those steps can be sped up if the body needs those red blood cells in a hurry. And also, those reticulocytes can be released early, the earlier stage of development of the red blood cell. So those red blood cells can be released before they're completely mature into the peripheral blood. So when we produce red blood cells, we also must make hemoglobin, because our hemoglobin is located inside our red blood cells. So what are the ingredients or the substances needed to produce more hemoglobin? Well, we first need iron. Iron is a metal. We need some vitamins such as vitamin B12 and folic acid. We need amino acids because hemoglobin is a protein and amino acids make up a protein. And we need hormones. The kidneys must produce erythropoietin to make more red blood cells and hemoglobin. Let's real quickly look at the structure of hemoglobin. Remember, our hemoglobin is located inside our red blood cell. You can see there's iron. Also, the hemoglobin molecule itself is a protein. We'll be looking at the hemoglobin molecule a little bit closer later on in another lecture, but this is just to give you an idea of what it looks like. It's made up of 4 chains, but again, we'll look at it a little bit closer in another lecture in the future. This is a critical thinking question that kind of sums up what we've been over today in lecture. And if you really think about it, you should be able to come up with the answer. I know the answer is right here, but try to think about it before you read the answer. And think about the information that we've learned today. So, would a patient on dialysis due to end-stage renal or kidney disease be anemic? And if you don't know what dialysis is when a patient gets hooked up to a machine and their blood gets taken out and cleaned and put back in, and they're basically doing that because the kidneys are no longer functioning and doing the work that they're supposed to do, because your kidneys usually clean up the blood. So that's basically what the dialysis machine does. It's cleaning the blood for the body because the kidneys are no longer doing it. An anemic is a low red blood cell count. So would a patient on a dialysis be anemic based on the information that you learned today? The answer would be yes, because a renal patient or a kidney patient are often anemic because their kidneys no longer fully function. Again, that's why they have to be on dialysis. Their kidneys are not functioning. So if their kidneys are not functioning, they cannot produce the hormone erythropoietin. And remember, erythropoietin is what stimulates our red blood cell production. So if our kidneys are not functioning, then our erythropoietin cannot be produced. Therefore, our erythropoietin is not going to be telling our bone marrow to produce red blood cells. So if our red blood cells are not producing, or if our bone marrow is not producing red blood cells, then we are going to be anemic or have a low red blood cell count. And you could even take it one step further. If you have a state of being anemic and you have a low red blood cell count, what does that red blood cell do for the body? What's the function? It carries oxygen. So if you don't have the red blood cells, your body's not going to have the oxygen, so you're going to have a state of hypoxia, low oxygen. This concludes our lecture material for hematopoiesis and erythropoiesis. Please make sure you're looking at your atlas to see those images of each step of erythropoiesis, and I will see you over in the next lecture.